Good morning, good afternoon, or good evening, depending on where you are in the world, and welcome to the start of MDDI's Focus on Fundamentals. It's a master class on reusable medical device cleaning, disinfection, and sterilization validations. This 3-day webinar series is sponsored by Nelson Labs and broadcast by Informa. Today is day 1, and we'll be discussing a complete guide to cleaning validations. My name is Peter Krass, and I'll be your moderator today. Before we get started, I have a few very quick announcements. First of all, this webinar is designed to be interactive. There's a dock of widgets at the bottom of your screen, and that will allow you to learn about today's speaker, download resources, and participate in a Q&A session we'll have toward the end of the presentation. The slides will advance automatically throughout the event, no need to click. And I'd like to ask you to please be sure to click the Register Now button in your viewing window. That way you can join us for the next 2 days of this Focus on Fundamentals series, when we'll be discussing disinfection and sterilization validations for reusable medical devices. And finally, if you have any technical issues during this webinar, please click the Help widget, that's at the bottom of your screen, or you can type your issue right into the Q&A area, and either way, we'll be happy to offer you one-on-one assistance. All right, let's move on to our day 1 presentation, a complete guide to cleaning validations for reusable medical devices. Discussing today's topic, we have Alpa Patel, Director of Scientific Improvement at Nelson Labs. To learn more about Alpa, you can visit the BIOS widgets that's at the bottom of your screen. With that, I'm going to hand it to Alpa Patel.
Thank you, Peter. Welcome everybody to this webinar series for learning about cleaning validations for reusable medical devices. For the next 45 minutes, I'll be going over all the criterias and updates on these validations and how they are represented in different regulatory platforms. So with the agenda, we're going to talk about understanding what is a reusable medical devices, categorizing of these reusable medical devices, how they're categorized in different regulatory authorities, or even in your facility, what are the criteria for those? Validation strategies used in market for these devices. I will go over a fishbone concept on how to determine what test methodologies are required for these validations. Requirements, as you know, that there has been a huge move towards these validations. So there are a lot of requirements out there that are probably not heard of, and there are still with question marks that are they really requirements or are they recommendations? So I'm going to go over those. And what is next for cleaning validations? It's still a moving target. There's a lot of things that we haven't yet talked about or even looked at that I will be talking over. And what do manufacturers have to be ready for when they're doing their submissions to regulatory all over the world regarding these validations? First, I'm going to go over reusable medical devices. These are devices that are used on patient over and over again, but between patient uses, they are cleaned, disinfected, and or sterilized. And to ensure that there's patient safety, that is a requirement that needs to happen. So these are just devices that are reused in healthcare facilities, home care, dental, anywhere you go. If you see a reusable device, you're sure to know that there are some instruction for use for them. Oops, I went too far, I apologize. When I talk about reusable devices, we're also talking about devices that are in the healthcare facility, right? Now you'll see these orange spots that are on the slide deck, where you'll see these devices are often utilized as healthcare practices, decontamination processes, right? You'll have AORN or SG&A or organizations that have written good hospital practices for cleaning and disinfecting instruments. And manufacturers usually are not involved in this process. But we're starting to see more and more discussions and more requirement on these non-critical devices that are now being asked to ensure that there is some sort of validation that has been performed and there's efficacy to show that there is safety, right? The two words that FDA likes, safety and effectiveness of the reuse of a device. So those two are very important. And so hospital practice now is being challenged with IFUs, instructions for use, for medical devices that are in their facility to ask manufacturers, do we have instructions for use for this? Because now we're being asked by joint commissions, or when we're being audited, what are the instructions for use for these type of devices? Now you might see that there's a light, there's a huge light, and these lights, I mean, the picture doesn't do justice. They are humongous, and we have validated everything I put here. We have validated before. The only thing I don't have here is a biohazard container, but we've validated those as well, as they are reusable and can be potential contamination to a patient through cross-contamination. So we are seeing a lot of these type of devices come through for validations as well. Now, if we look at what sector of the industry is reusable, it's not a huge portion, but it is starting to become a huge portion of the industry. We often are linked with single-use devices you use, throw away. Now we're starting to see those single-use devices migrate towards reusable and being asked, can these be reused? And so we're seeing med device industry trying to make devices that are single-use now reusable. So we have a huge sector that we're working with. Surgical instruments, orthopedics, ENT devices, dental, robotic, capital equipment that we just talked about in the previous slides, all flexible endoscopes that we work with. Dialysis machines are something that we've seen in the last 5 years take off, right? Those are devices that were not validated. They had little human factors validation performed, but not disinfection and cleaning efficacy on the exterior and the internal portion. So we're seeing those kind of machines come through, automated cleaning reprocessors. Yeah, we do validate flexible endoscopes, but we also have to validate the machinery that's actually cleaning those. So those as well. And we're starting to see clinical studies come through as well, stuff that's used in the hospital to make sure that they're cleaned. So we're seeing those clinical tests and home care products, baby glucose, anything that's home care, even insulin pumps, where CPAPs, those kind of devices also require validations. And we're seeing an uptake on home care products now. One thing I like to talk about is we always make sure that when we're writing protocol or we're designing cleaning validations, we keep in mind the central sterile processing departments or sterile processing departments in hospitals. We're validating what's happening in the hospital, right? Manufacturers give us an IFU. When we're using the IFU, we're validating the IFU, but we also have to keep in consideration that these IFUs are going to be followed by healthcare. And so when we design a protocol, we're designing how the devices will be used, how they will be bought in, and what steps are going to be taken. So we always want to consider the layout of a central processing department. And you'll see here, this is the dirty area. This is all I'm going to be talking about today. It's this dirty area. Once the devices come through those doors into the sink and into the washer, and you can see that there's doors on one side and on the other side. It opens up on those and it goes to the clean side. And then it goes to the sterile side. So today I'm talking about the dirty area. And tomorrow they'll be talking about disinfection and then sterilization. So I just wanted to give a visual of what we will be discussing today. Validation of reusable medical devices. This is a nutshell of all the validations that you must consider in your design history file for anything that's reusable. You have to have a cleaning validation. You have to have a disinfection validation, chemical or physical. You have to have a sterilization validation. And life cycle testing is something that is being discussed currently. And there are some caveats when it comes to disinfection, which we'll hear tomorrow from my colleague. But it is something that should be part of your portfolio and why it's not being performed. Now, if you look at the process in a healthcare setting, you will see point of use processing. That is right at the bedside or after surgery, where they're putting the instruments into some sort of a container that will be taken down to the reprocessing area to clean all those devices. So that usually is not part of the cleaning validation. We omit that point because not all hospitals actually perform it the same way. And although the instructions for use will say clean immediately, we know that that doesn't happen. So that is always omitted from validations, but thorough cleaning, sterilization, and disinfection are the three pillars of validations when it comes to reprocessing validations. I'm going to go over cleaning validations for this part of the webinar and walk you through what are some of the studies that you have to do within the cleaning validation. Here's a fishbone concept of how we start with cleaning validations, and I'll be talking over simulated use cycles. What are those cycles? Why are they required? Then I'll jump into cleaning validations. How do we perform them? Test soil selection. Are detergents important? Is water important? And what are the methods for these validations? And then we'll go over extraction, right? After you clean, you have to see what's there. So we're going to talk about analyte testing. What is required for analyte testing? And they're different in different countries. What are the main analytes that are tested, right? Protein is one that's always tested. When do I test TOC? When do I test hemoglobin? When do I test carbohydrate? Are those things that I should worry about? And how do I report these results to the FDA? What are they looking for? So those are the things that I will be going over today. Now, I wanted to bring this to everyone's attention. This is standards. These are standards that are used to design a cleaning validation. If you look at them, and if you look at the years, just past few years, they've been updated. And some are new and some are old. But SD98 is the first standard that is written for how to perform cleaning validations. Okay, so it is to design a cleaning validation and what you need to have in the design for the validations to be successful for submissions. So that is what's written in the standard, is it helps you design the validation. So if you're not familiar with the standard, I would suggest that you do get familiar. It was published last year in August. But there's a lot of interpretations in there as well. It doesn't tell you exactly what to do. It gives you how you should go about doing them, but it would be between the manufacturer and the laboratory to design the validation plan by itself. But these are a lot of standards. I've only put a snippet of it. There's still probably another 15 that are still being developed and are still being currently worked on just for reusable medical devices. But these are the main ones that have been updated to bring attention to why cleaning validations are so important for reusable medical devices. Now, we just talked about AAMI SD98. This used to be TIR 30, if anybody remembers. It started a long time ago. We had TIR 30 in 2003, and then it moved, and it took different revisions until it was decided a couple of years ago that we're going to make this into a standard, and it became a standard after a lot of debate and a lot of arguments, believe it or not, between FDA, industry, and lab, and consultants and third party that were involved in these decisions. But the main changes from what the TIR said compared to SD98 is that there was just more clarification to what needs to be tested. And in these clarifications, you will see that TIR 30 did not have simulated use cycles, right? They didn't talk about that, but FDA guidance document did. It was 2011 version, and simulated use didn't come out until 2015 when the FDA guidance document for labeling and validating of reusable medical devices was published. Once that was published, the word came in, simulated use cycles. And so that is something that was not there but now is in SD98. Sample size, we were always historically thought that 3 is good enough for reusable medical devices. However, SD98 has taken a different approach and has given the manufacturers to decide what is the right sample size for your product, depending on the complexity and the reusability of the product. So you just have to look at that. And also criticality plays a huge chunk in how to determine a sample size. So now that's left into interpretation for the manufacturer to decide. Positive controls have increased. We only did 1 positive control. Now it requires that we do 3 positive controls for semi-critical and critical devices. And these devices, the ones that are penetrate through the skin into the bloodstream for critical and semi-critical mucous membranes. So those ones you have to do 3, but you can still do 1 positive control for the non-critical devices. Extraction efficiency was not specified. Now it does specify that you want to show a greater than 70% recovery. It doesn't say you have to. It says it's recommended and gives you examples of why it's appropriate to have the 70%. Non-critical devices, this is a plus for everybody, where we used to do analytical tests for non-critical devices. And these are devices that I showed you in my previous slide, where there were surgical lights and stools and bed IV poles. So those we used to do protein and TOC. However, it's not required. I mean, when we were doing it, we also thought that this is overkill, but there was nothing written that you couldn't do it. So everybody was doing it anyway. But S98 brought light into it, and although they have 2 analyte markers to be tested for critical and semi-critical devices, for non-critical devices, you don't have to do any analyte testing anymore. And simulated use is not required for non-critical devices. You can just do your testing without the same uses, and you can just do visual inspection, wipe it, it's clean, it's good to go. So that is a big, huge change to these type of devices. So it is a plus change for the manufacturers because cost has gone down and it doesn't cause a lot of problems. But for disinfection, it's still a juggling factor for us, which you'll hear tomorrow from my colleague. Final results were reported as micrograms per centimeter squared, right, in TR30. SD98 is the same thing. You're still reporting in the same manner as cleaning validations were reported before 2022, right? The only thing is that it does specify that you may want to apply a standard deviation to the analytes, which I'll go in detail once we get to that slide. Now, we talked about simulated use cycles as one of the changes that happened in SD98. What I'm talking about is these simulated use cycles are cycles where you are making your device into a used condition of how it will be used at a healthcare facility. So if you think about your devices in the hospital, they're being used on a patient, then they're being cleaned, and then they're being sterilized, right? So that's exactly what we're doing for simulated use, where we're doing the soiling, the cleaning, and the disinfection or sterilization, depending on what your end processing point. And then we're doing a visual inspection to see if the cleaning is adequate. And over time, in 6 simulated use cycles, do we see anything that's retaining? So it's very important that they brought this into light, because if you think about it, if I got a device from the manufacturer that was nice and ready to go, shiny, it's beautiful, it will pass with no complexities. But if you think about your devices being in healthcare and they're being reused so many times, are they still cleanable at that point? So this is what the FDA is trying to bring attention to, and that's why it was added to SD98 to make sure that these simulated use cycles are being performed. Okay. Now you might ask, where's the 6 come from? That has changed over time. When we first started in 2015, we only had to do 3 simulated use cycles per regulatory recommendation. Then it went to 10, and now it's 6. So it may change in the future, but for now, they recommend to perform at least 6 simulated use cycles. They don't specify you have to do 6. They say we recommend you do 6. And we've seen regulatory move towards if your devices have a shelf life, or use life of 10 or 15, then we would probably do 15 simulated use cycles. It's not that far off. So we would probably do that because they have asked customers that have said that their use of their product is 10 times and they did 6 simulated use cycles. They said, no, you need to do 10 and show. So we have seen that before, okay? So this is not, 6 is not set on stone, but majority of the devices does go through that. Now with this is, it just hits to heart why simulated use cycles are important for validations. This is a picture on the left where debris was retained in surgical suction tubes over time, right? When you do the validations, you don't see it, but over time, you start to see that tissue gets stuck when you're brushing and when you're flushing, and those areas are not accessible. And that caused an outbreak, and they had to recall the devices. Now, on the right, you'll see rusting of internal lumens after 7 full processing cycles. So when you validate studies, after 6 cycles, everything passes and everything looks great. And on the 7th one, we do the validation and it looks great. Protein passed, hemoglobin passed, but residuals failed, right? So when I looked into the device, it was pretty clear that the device is pretty much tarnished from the inside, and that's what caused the cells to die when we did cytotoxicity testing. So regardless of them passing all the analyte testing, which would have been fine, the device from the internal was destroyed. And we had to report back to the sponsor and tell them, listen, your devices have passed everything, they look great, but the internal portion is destroyed and the detergent residuals, you failed. So you would have to go back and redesign the internal portion to make sure that they can withstand the chemicals and the water that's being used to clean these devices. So something to think about, just food for thought. Sample size and controls, of course, has dramatically increased from what we were usually performing for these validations, as you can see on the slide. We have positive controls that increased. We have to do positive sample controls. Test article samples increased, negative device controls, negative sample controls, and cytotoxicity testing to assess detergent residuals. If they're residuals after cleaning, what do we do with those? So that all of these controls are not a recommendation, they're a requirement. You have to have all these controls in place to show that the validation is done accurately and the measurements that you're getting are accurate. So these were already in 2015, but because FDA's documents are usually recommendations, not a lot of companies were doing all these controls, but with the SD98 update, these controls are now mandatory. Usually we get a device looking like this, right? You get a tray for reusable devices. Say we get an orthopedic tray, and they'll be like, okay, let's do cleaning validation on this. So you'll be like, okay, what do we test from here? And for sterilization, you would do the entire tray. But for cleaning validations, we can't do the entire tray. We would have to validate them, each one, or group them in groupings. So you can do that as well. So we would pick a worst case device out of this selection, and we would do a validation on that worst case device, and showing that if you can clean this worst case device, you can clean the rest of the devices. So you have to choose the right one. Now, don't get me wrong, you can't choose one. You would have to choose different features, different materials, lumen sizes. A lot of things play in what is a worst case device for cleanability. And one thing to remember is worst case for cleanability is not worst case for sterilization. So you have to keep that in mind. So from the previous slide, I would choose this because it's got knurls in there. I can open, there's rotations everywhere. And if you look at this one, you can see that there's screws in there and all of these features, the knurls right here, in here. How do I clean this device, right? Those are the features that are very difficult to clean, and that is what we would look at and use that. Now, family grouping is also a great way of doing validations for reusables. You can always choose devices that look similar and group them in families depending on the materials, the size, challenging features, and how they're utilized during use. So that important factor is you can either choose worst case device or you can do family grouping as well. And a lot of people do family grouping where they will have the worst case device and then they'll group their families in there for testing. Difficult designs, they're all out there, right? We don't like to see them, but you all need these designs. I mean, materials and designs to work together with reusables, right? Braided cables, we can't get anything off of them. Those are the hardest things to clean. Aluminum-based metals, they destroy very fast and tarnish with a lot of chemicals. So you have to be very careful with that. Silicon rubber O-rings, they break apart really quickly because detergents get into them. They're porous. And so they'll get into them and they'll start to break them apart. Textured surfaces hold on to test soil, bodily fluids, and tissue and things like that. Dead end lumens, no device should have a dead end lumen. You have to have a way of cleaning through, right? I can flush all you want, but once it's stuck at the end of that barrel, I'm not going to be able to pull that off, no matter how many brushing steps I have that. So these are some of the things that have caused problems before in cleaning, but they're not avoidable. We need these in there to function, so it is important to have them. One thing that regulatory talks about a lot is disassembly, right? If you have these features that are worst case to clean and are very difficult, can they be disassembled? And if they can, how are you disassembling them? And is it simple enough for the users to understand how to disassemble? You know how to disassemble them, or engineers know how to disassemble because they designed it. You give them to me, I may be able to figure it out, but you're giving it to the healthcare users that don't have the education level that either the lab, or the manufacturer, the engineers that are working on them. So you have to break them apart like the way this has been broken apart. It's very well defined and it's in the IFU. So we see this a lot as well. And a lot of times manufacturers like to validate them in a non-disassembled, just in case they forget to disassemble. But oftentimes with these type of designs, we do ask for disassembly. Selecting sites are a big, it is a big issue because we want to make sure that we are challenging the sites that will be contaminated during use, right? A lot of times we see a lot of disagreement saying, no, this will not get soiled. Let's just not worry about this. But we usually would recommend the areas that we often see devices, and we see thousands of different tested devices that flow through the lab for validation testing. So selecting sites is something that we will work with the manufacturer to design and figure out what needs to happen. But there are some that you don't have a choice in, right? Like for flexible endoscopes, you can't you don't have a choice in that. FDA has made that choice for you. And these are the ones that you have to validate. Control handle is something that you may or may not want, but majority of the time we are selecting test sites that will be contaminated during use. And we would take it to a worst case approach at that point. Now selecting simulated use soils, right? Now we selected your sites. Now we have to figure out what am I putting on this test sites, right? And we have to make sure that we're using the right simulated use test soils. And Anything with blood, you will see coagulated blood, defibrinated blood. If you see mucous membranes, you see different types of sebum or British test soil, or you could even do a mixture. Okay, this is left to your interpretation. How your devices are being challenged is how we are going to validate it. If you say that we want you to add bone to it because it's an orthopedic device, we want to see bone, we would add bone meal to it, or we would try to get a pig bone and drill in it and get all that out and put it in the test soil, right? We can't do everything, but we try to simulate as much as possible as we can. And we're seeing regulations really asking and honing into this simulated use soil, right? ST90 goes in very detail that things that are used in procedure with the devices, you want to add that in your test soil to make sure that they can be removed adequately. It's very important that we have that understanding, because we have seen validations come back, and especially dental. You would think that dental devices are simple, easy, a little bit of blood here, we're good to go. But because they're going in the teeth, they want to see bone added to the test soil and being challenged. So some of the things that we have not done previously or even thought about, we now have to think about them. And engineers have, because we don't know the device, we don't know how it's used. We're looking for recommendations from the manufacturer to understand, how is this being contaminated? What is left on the device after the procedure is done, right? And that's the important part that you have to figure out. For example, cauterizing devices, robotic devices that go in and there is electrical energy, the blood is cauterized on the end of the bipolar forceps. That has to be mimicked in a laboratory environment. Now, you just can't, and then you have to subject that to the validation. So we are starting to see movement towards more surgery type of simulated use test soils than what historically was used. This is very important to have, because this is how the clinically relevancy of the test soils have to be maintained and discussed in detail, right? It's important that we have what is used and how is clinically relevant. FDA and any regulatory body would like to see that. So make sure you have this chart available. Soiling methods. After you've selected your test soil, you have soiling methods, and this will help us understand what soiling methods you have to use, right? There's all different types. You flush, full immersion, partial, whatever method we choose has to be what is being performed in the surgery suite or is performed in the clinical procedure. And then we will take a notch up to make it worst case. So that's why we've got combination of methods here. And it's always determined through contamination method, how it's used, how it's simulated in surgery, what is the soil time, how long it is used, and then at what portions do we, once it's done, how long can it sit? So those are the things that we consider with manufacturers when we're designing a cleaning validation for these devices. Now, this gives you a good example and a visual of, you can see that the hemostats are just being used just on the probably 2 inches of the proximal end, and just to help tie the suture. But if you see how I've soiled it, it's probably all of it. So it's very important that in case it falls or it gets more dirtier, we have validated the worst case, right? Obviously, we don't want to overdo the worst case, but there is that factor that we do take in for validations. Soiling methods for robotic devices, like we just talked about cauterization. This is a cauterized bipolar forceps, so you have to worry about that. And then the shaft would be handled through handling. So these are the things that you have to consider during validation. And soiling methods for flexible endoscopes, right? If you see the the light guide is connected to the computer, but if there's a backwash, ever there's a problem, this portion might get contaminated. And so that does get simulated in actual laboratory testing as well. Now, dry time for cleaning validations is something that has been discussed very, very, very historically. I started these validations 20 years ago, and dry time was different to what it is now. And what we did before was we just place a wet towel over the device and keep it moist. So it's moist when it goes down for reprocessing. Now it's a different story. You know, dry time is simulated use where we're waiting till it's dry, right? You see a picture here, and this picture actually is a true representative of what it would look like. So we do validations where the device is dry to the touch. Methods of cleaning, there's manual, mechanical, and automated. Manual is just a soak and a brush. Sonication is right over here, and then the automated cleaning is we're using a large washer to clean these devices. Now, one thing to remember with automated cleaning is when you're validating a tray, you want to ensure that the tray, is it being cleaned? Are your devices being cleaned in a tray or outside of the tray? So those are the things that I would focus on and bring into attention, because a lot of times we get devices out of the tray for validation, but we don't know if it's being put back in there in the hospital for use when it's in the washer. So all these things have to be discussed ahead of time to design a validation plan for automated cleaning. One thing to remember is for automated cleaning, the thermal phase of the cycle is part of your IFU, but we don't use that for cleaning validations because that is a disinfection validation. So that cycle will be omitted from the cycle, from the cleaning validation. It's only used for simulated use cycles because we are mimicking what's happening in the hospital practices. So something to keep in mind when validating these devices. I look at validation parameters. Here you can see manufacturer's IFU, and then you see validation parameters. We will always take it to the worst case, okay? So soiling will be worst case where we're challenging the soiling, but cleaning will take it to a worst case where we're also challenging the cleaning by using the less dosage, the less time, if their volumes, temperature, everything will be less than what your IFU has specified. Now, not everything will be worst case. You have to determine the critical steps that need to be decreased for the validation. Detergents are something that's very important to understand because detergents are what cleans your devices, and some are caustic and some are not. So you want to make sure that you're using the right detergents for your validations. And there's neutral alkaline and acids. Like for Europe, they use alkaline detergents that can be very caustic to your devices, but they are followed by a neutralization phase. So you have to understand that there's a neutralization phase for that as well. US predominantly use neutral pH detergents. So that is a must for US as well. So there are these balances that manufacturers have to decide on material of your product if they can withstand these chemicals. Now for detergents, for critical and semi-critical devices, you would use enzymatic detergents, and it must have protease in it. It has to have some sort of enzyme to help protein remove off of the devices. So that is what we're looking for when we're doing these validations, right? For non-critical devices, it's a wipe basically. We're using a wipe, or we're using a spray, and we're just wiping the devices with it. So these are the differences between the two classifications of medical devices that are used for validations. Now, the detergent chemistries, you will see that there are different types of detergents, and they all have different types of enzymes depending on what the target of that detergent is. But this is just a visual demonstration of all these different enzymes that are in detergents that you may have never thought of. when you're doing these validations, but something to give you a visual depending on what kind of enzymes we're looking at when we talk about detergents. And some of these enzymes can cause problems or can be sticky to your devices, which can cause problems with the analytes or cytotoxicity testing as well. So you want to make sure that these enzymes are okay to use as well. Rinsing methods of rinsing, as if you, the standard AAMI standard just got updated, TIR 34 to SD 108. And now these are water criteria used for reprocessing. And it's just some of the things that you may want to be aware of when it comes to rinsing, right? We have utility, critical, and steam. And utility water is what just comes out of the faucet. Critical water is something that has been treated. Some sort of treatment has happened to the water. But for cleaning validations, we're talking about utility water. We're focused on pre-rinsing, making your detergent baths, flushing the devices, cleaning. The only thing that we think about critical is the last phase. And rinsing has to be done with critical water. You don't want to do all this cleaning and then use dirty water to clean your device and make it dirty again with bacteria, right? So that's the reasoning why we have critical water at the end of processing. This is material compatibility. It's not just cleaning off residuals, it's also material compatibility. If you use the wrong or poor quality water that have a lot of heavy metals or elements that can cause pivoting, corrosion. This is pictures from our automated washers. These are devices that are used as dunnage, devices that we put in the automated washer with our devices that we validate. And over time, these are over years, actually. Over years, this is what's happened to them, right? Although we're using utility water for cleaning, the last cleaning is done with RODI water. And even with the last rinsing with RODI water, over time, this is what it's done. So it's just something to keep in mind, that the water that you used for cleaning your devices are very, very important. We in the lab will make sure that we're using the right water. It's the manufacturers have to make sure that when they're selling their devices to the hospitals, they have the right water quality for your product. Once all the cleaning is done, we're doing a visual inspection, right? It's required that you have to have a visual inspection. If we see soil after cleaning, you fail the test and you have to repeat it again, or you have to increase your rinsing steps. There are mitigation steps that you would take to do that. There is 10x magnification for duodenoscopes. There's borescope recommendations as well, but those are just recommendations for lumens. But visual inspection through normal lighting conditions with the naked eye is a requirement. Once they're visually inspected, they are extracted. And there are many methods of extraction, but sonication and manual shaking is one of the methods that we use. Swabbing is the last thing we would do, but those are for non-critical devices, right? Devices that I cannot immerse, and they're large. And so that is the last one we would worry about. Once you have extracted them, now that extract fluid is going to be tested for analytes. And these would be protein, hemoglobin, TOC, all of that stuff. And you have to test two. It's required by SD98 to test two clinically relevant markers. Protein will always be tested. And then hemoglobin and TOC. If your devices come in contact with blood, you would do hemoglobin. If your devices come in contact with mucous membranes, it would be TOC. So you just have to determine what's appropriate for the contact of your device and what it comes in contact with. Cytotoxicity is a recommendation, but we see it almost as a requirement now, that you have to analyze that there's no detergent residuals left after the cleaning validation. And that's done through a series of different tests, and you can decide what test is appropriate for the testing. We usually do MEM, but there is MTT and TOC. TOC is very good candidate for that test as well. These are some cleaning validation acceptance criteria. If you see that AAMI SD98 has their criterias for all of these, and then we have ISO 15883. And you will see that they are the same for action, but there's an alert level. And what does this alert level mean? There is no meaning to it. It's just that it just alerts you that when you're at that, you may want to consider looking at your IFU or your reprocessing instructions to make them more stringent. But that's all there is to that. Now, one thing to remember is there are different criterias in different countries. And so you want to make sure that you're using the right criteria for the right country. And protein is one that is very much already outlined, that Germany, France, Netherlands would want to see a 3 micrograms per centimeter square for protein compared to 6.4 micrograms per centimeter square required by FDA. So that is where you will see the difference. So when we do validations, we probably want to go to the lower end so you can sell your product everywhere and it's within the regulations. This is the example I was talking about that was in SD98, where standard deviation is used to determine if your results are valid. Now, not enforced by regulatory, it is recommended that you look at that, but regulations have not enforced that yet. We haven't heard from TÜVSÜD, or we haven't heard from anybody yet about or FDA about this. That's been in SD98. So we're just doing what's required currently, but it is in the document. So I just wanted to bring your attention to that there is this standard deviation between the test replicates that you may want to consider, and you add the standard deviation to the highest value that you got, and if that is above 6.4, like I've highlighted here, then you have to test with a higher sample count. And so that's outlined in SD98, but not enforced yet. Now, cleaning validation is done. Now we talk about how do we determine material compatibility with these detergents and with these soils and all that stuff. Is end-of-life testing needed? Are they linked to healthcare-acquired infections? Pretty sure they are. How does manufacturer know that their devices are breaking down? You don't know until you test them. And this is an image that was, it was in the news many, many, many years ago from a study that was conducted at Michigan Health System. And they saw through that this suction device was only used like 20 times. So you could flush through them. But if you put a brush through, you can put a brush through them and you can see how much soil was retained in them. So right now, the understanding here is do we need to do these testing or not? And if you look at ISO 10993-1, 4.8, it says for reusable medical devices, biological safety shall be evaluated for the maximum number of validated processing cycles by the manufacturer. So we're seeing a push from EU using this ISO standard to have you determine biological safety of your reuse cycle. So we're starting to see these end-of-life use testing creep up, and we're starting to see testing being performed on these devices now. It may be 50 cycles, 100 cycles, or 200 cycles, but we're starting to see them because they are being discussed, and they are currently being discussed right now to even make it more stringent about that you have to now do it. I mean, it says shall here, so it is a requirement, but it hasn't been enforced readily. The good news for manufacturers is that the ISO FDA do specify that MDM must let the end user know when the device is no longer usable. As long as they say that and train them or write them in the IFU, you don't have to perform this end-of-life use testing. So here's the leeway, right? Some ISO says you have to. ISO for cleaning says you don't have to. And the guidance document says you don't have to perform them as long as you let the user know when the device is no longer usable. Sorry. So I'm leaving you with that in your thoughts. And these are the guidance documents that I used for this webinar. So if you want to look them up, they're all for reusable devices. And that takes me to the end of this webinar for cleaning validations. And I would like to hand over to Peter for any QA for the Q&A session. Thank you, everyone.
Thanks, Apple. That was really good. Before we begin, everyone, the Q&A, just want to remind everyone about the webinar survey. It's available on the right of your presentation window. If you've closed the survey, you can reopen the widget. Just click the icon along the bottom of your screen. And we hope you'll fill out the feedback form before you leave us today. Your participation in the survey can help us to serve you better in the future. All right, we've got a lot of questions, which is great. If we can't get to them all, we'll certainly share them with Alpa, who can reply to you offline, but we're going to do our best to get to as many as we can. Here's a first question, kind of basic. What is the difference between disinfection and sterilization, if any?
Okay, that's a good question. The difference between disinfection and sterilization is the criticality of the device, right? Which you'll hear tomorrow. If you are using semi-critical devices, they are subjected to disinfection. These are devices that come in contact with mucous membranes, right? But if they can be sterilized, they should be sterilized. So the difference is, can the devices be sterilized? If they can, yes. If they cannot be sterilized, then you have that mode of disinfection. A lot of respiratory devices cannot be sterilized because of material compatibility. So they have to be disinfected. And so that's when disinfection would play a role in.
Okay, great. Here's our next question. How final are the test articles expected to be? In other words, in a development program, is design equivalent acceptable as opposed to production equivalent?
So because these are validations that are going to be used for submissions, they have to be in final configuration. You cannot, it is okay to not have electronics that are internal components. We have seen devices with electronics omitted, that is fine, but the product will have to be in final configuration because cleaning and disinfection for reusables or sterilization is a requirement. And the FDA will definitely ask for a final product configuration. Back when I started in 2003, you could use a device that looks similar to that and then just say that it should work the same way. Not anymore. Now they want to see final configuration.
Okay, good. Here's a new question. What are the 3 positive device controls as opposed to the previous requirement of 1? In other words, how do you obtain them?
So what the positive controls are is they're validating that the extraction method you used is adequate, right? And you did remove enough. And 3 is historically known as a number that's used for reproducibility. 1 only gives you that at that moment, this is what we got, but 3 gives you reproducibility of the extraction method, that you were able to remove whatever there was on the device. And so 3 helps the regulatory and even test lab to understand that, okay, the extraction method that was utilized to extract these devices was adequate. So that's why they moved to 3 positive controls. These are devices that are contaminated, not cleaned, but just extracted. So they contaminated and extracted.
Okay, good. Next question. How are non-critical devices defined? Non-critical.
Non-critical devices are defined as devices that come in contact with intact skin only. So devices that are just sitting like blood pressure cuffs, or devices that are sitting in the clinical facility or OR room that is supporting critical features, right? So if you think about robotic arms, the robot arms are critical devices, but the machinery that's holding the arms and moving them around are non-critical. So they will be validated differently than how a robotic device would be validated.
Sure, that makes sense. Okay. Here's a question about discarding. We talked about this a little bit. When does one discard a reusable medical device? And are there any criteria you can use to, make this decision?
The only way you can make this criteria is by testing. We can't tell you when your device can be discarded, right? A manufacturer has to know at what point does the device stop functioning. And as the only caveat here is, there is no one detergent that's used. So when you do these end-of-life use tests, we're using a detergent that's commonly used. However, you go to Germany, it might be something different. You go to Australia, it might be something different. You go to Africa, it might be something different. So to try to assess all these different chemistries that you're using, you don't know. So you have to do some sort of R&D test, either internally or with a third-party laboratory, to establish what is the life of your product. Manufacturers have to know when the device is no longer usable, and they have to notify the users or through the IFU when it's no longer usable, and you have to either throw it out or contact them for a replacement.
Okay. A related question here. How about life cycle end-of-life testing? Does it have to be done including soiling the instrument? For example, if the IFU says 300 cycles, is this done or required?
If the IFU says 300 cycles, it is 100% required. Huh. So before you write a number into your IFU, you want to make sure that you have data supporting that after 300 cycles, your device is still cleanable and safe to use on the patient. That's why we don't see a lot of people write numbers in there, but they do side tests to ensure that after 100 cycles, we're still seeing levels that are below for the cleaning validations.
Okay, here's the next question. If our product requires sterilization before using in a patient, but that must be handled by the hospital and not by us, manufacturer, should we still include the clinical relevancy in our cleaning SOP?
So if you are doing a cleaning, these are single-use devices that are given to hospital to clean and sterilize beforehand. That would be something that it is a gray line there, but yes, we do validations on those. You have to have some sort of testing performed to show that after cleaning, the residuals of detergents are not there or impeding any sterilization practices.
Okay. Next question. During the design stage, how do we decide what level, to what level we need to disassemble the device for it to be reusable?
That's a very good question. When you start to see that there are areas where test soil is going to go, or bodily fluids are going to be trapped, that there is no accessible through flushing or even brushing, that's when you have to start deciding that we need to we will have to pull it apart to make sure that those areas are accessible.
Okay. We have time for another question or two. Here's one. Is there a mandate or guideline on sample size for cleaning validation? I have a non-critical device. It's on the larger size and fairly difficult to make. So I'm curious to know if I should prepare for a number of devices or if it's possible to use a single device or a small number and then take multiple sample sites to gather more data points.
Very good question. Yes, we have had many non-critical devices that are huge that we only get one sample of, but we have to perform 3 replicates of the same sample, right? So you can submit one sample for testing, and we would use that sample multiple times to get the replicates necessary for the submissions. And usually for non-critical devices, 3 has been okay to use.
Okay. Let's see if we've got any more questions coming in. I'm just taking a quick look here. Oh, here we go. Could you clarify the sample size and controls slide? Could you clarify that?
Yes, absolutely. So the sample size and controls slide, if you look at it right now, I have 3 samples for positive control. Now the positive sample control is where we're testing the test soil can be analyzed after it's been extracted. If you think about it, after cleaning, you're extracting your devices. I'm assuming that after cleaning, I've removed everything off. But if there was even a slight chance of protein or hemoglobin in there, can I still detect it with the chemical assays that we're using? And that is what the positive sample control tests. Test articles are devices that are inoculated and processed. So these are subjected to what you think would be appropriate for your product type, depending on criticality and complexity of the product. Negative device control is just processed, right? So it's processed to see if there's any residuals that will impact the chemical test that we do for these devices. Negative sample control is just a blank. It's just a water extract that shows that the assay that we did and the curves that we made were free of any interferences. And the cytotoxicity control that's just determining any detergent residuals that were left on the device. Is it safe? It's answering the safety factor for that.
Okay. Well, we're up at the top of the hour. That's all the time we have for questions. Thank you, Alpa. Any closing statements for our audience before we say goodbye?
No, I would just like to thank everyone for joining this series of the webinar for cleaning validations. If you have any questions, you have my contact information. Please contact me, and I'll be happy to help you with any questions you have. Thank you.
Thank you, Alpa. We appreciate your time and expertise today. I'd like to thank our sponsor, Nelson Labs, and I'd like to thank everyone out in the audience. We appreciate your attention and participation. Within the next few hours, you'll receive a personalized follow-up email. This will contain details and a link to today's presentation on demand. If you have colleagues or peers who could not join us today, please invite them to listen to this event on demand. This webinar is copyright 2023 by Informa. The presentation materials are owned by or copyrighted by Informa Markets, and the individual speakers are solely responsible for their content and opinions. On behalf of our guest speaker, Alpa Patel, I'm Peter Krass. Thank you, and we look forward to seeing you tomorrow.